few more pointers, and things guys do well. All right. Oh, boy, I already used those out. All right, so we're doing chapter 6, 7, and it says a little bit of 11, which deals with cell walls. I kind of overviewed these things a little bit. But we talked about already cell theory, plasma membrane, moving things across the membrane through diffusion, diffusion, active transport. We talked about the nucleus and what goes on there. We talked about cytoplasm, the membrane systems. We talked about the two different types of ER. We talked about the Golgi. And then we were talking about organelles. We talked about single membrane organelles. We started talking about and dealt with most of the double membrane organelles. So what are the two double membrane organelles we talked about? Mitochondria and chloroplast, right? So the mitochondria shown in figure 714. Again, there's two membranes that surround it. Here shows the inner and outer membranes here. And they're shown here with the outer membrane being kind of a purplish color, the inner membrane being kind of a bluish gray color. Right? So there's two membranes around there. On the chloroplast, same thing. Both of these are energy molecule or energy organelles. They have inner and outer membranes, not shown real well here, but you can see two membranes around the chloroplast. So the question I posed right at the end of the class on Monday is, where do these two membranes come from? Why do these organelles have two membranes, but things like lysosomes and peroxisomes and vacuoles only have one membrane? Right? All right. So to answer that question is we have to talk about what's referred to as the endosymbiotic theory of organelle origin. That's a mouthful. So to help you with that, it's referred to, I got up here, we're actually going to jump back to chapter 27 for just a minute here, look at one figure. Now, you don't need to read chapter 27 yet. We'll get to chapter 27 later. Figure 27.7, it just shows the, the endosymbiotic theory. Now, to help you with the idea of the endosymbiotic theory of organelle origin, explains how these double membrane organelles showed up. Okay? So, what does the prefix endo mean?
Inside.
Means inside. Do we know what the term symbiotic means? Or symbio? Yeah, shared relationship. So biotic refers to living organisms, right? Talk about something that's bio or biotic, that's living organism. Symbiotic means two organisms living together. That's a symbiotic relationship. Okay? They're living together for whatever function. Okay, so the endosymbiotic means two organisms living together, but one organism is literally inside the other organism. Internal. So it's two organisms living together, but one is actually physically inside the other. Right? And it's the theory of organelle origin. So if it's a theory, what does that mean? What?
It's not 100%.
Yeah, but let's be more positive.
It hasn't been proven incorrect.
Yeah, it hasn't been disproved yet, and why has it not been disproved? Mainly because there's extensive evidence that back it up, right? There's extensive evidence. We don't just simply come up with theories because we found one piece of evidence, right? There's extensive evidence for this. All right, so we'll talk about some of the evidences here. So what's the endosymbiotic theory really state, right? So how did it happen? Evolutionarily, we had what is going to be considered the host cell. Probably a eukaryotic cell. Doesn't show a nucleus in there, but probably it's a eukaryotic cell. Now, we know that cells will engulf other cells, smaller cells, right? We talked about that before. When we talked about the lysosome, we mentioned about what's referred to as phagocytosis. So one cell will engulf another cell and then fuse that cell with its lysosome, break it down, and get materials out of it. Right? However, the endosymbiotic theory states, and there's evidence for this as well, that the host cell surrounds and engulfs a bacterial cell. Okay, so this is a large eukaryotic cell, engulfs a small bacterial cell. But instead of breaking it down, that bacterial cell survives. Right? It stays inside the host cell. Okay? It survives. Now, the bacterial cell can do certain types of metabolism. Right? So in this case, that bacterial cell can take in things like pyruvate and oxygen, and it can generate ATP because this free-living bacterium was able to do that. Right? Take in organic molecules, break them down with little oxygen, generate some ATP. But now, since this cell is inside the host cell, it's doing the same thing, but it's releasing the ATP to the host cell. So the host cell actually benefits from this, right? Because the host cell now can generate more ATP because it's got this little structure that can do that. Now, what I want to do is look at some of the evidences for this. Let's go back and look at this idea of engulfing a bacteria, a prokaryotic cell. Notice when it engulfs the cell, it puts its own membrane around that cell, right? So when we look at that cell after it gets engulfed, how many membranes are around it? 2. Okay? That supports the double membrane. But you say, okay, well that's kind of loose evidence. But let's look at those membranes. Notice that on this, what we now would call an organelle, because this structure now is trapped inside that host cell. It's got those two membranes, right? So this now would be what we would call a mitochondria. Okay, that's the origin of mitochondria. We'll talk about chloroplast in a minute. So if we look at a mitochondrial membrane, look at those two membranes. What are membranes made up of? Phospholipids. What's a phospholipid look like? It's all purged that out of the membrane. Okay, that was on quiz 1. We don't. What? Okay, got the polar heads on the outside, and toward the outside of the cell and the inside. Okay, what are the non-polar tails made up of? Fatty acid. All right, made up of fatty acid. What's a fatty acid? What's a fatty acid? What's it made of? No. It's the subunit of a lipid, so it's not made of lipid. That's a dog chasing its tail. Subunit of lipid made of lipid, which is subunit of lipid. So, what is a fatty acid? What's it look like? If I said draw a fatty acid, what would you draw? Good. There we go. If I said draw the molecular structure of a fatty acid, what would you be drawing? A long carbon chain, right? We said they're made up of long carbon chains, anywhere from 16 to as many as 36 carbons. You can do about 16, 24, 26. All right. If we take a look at mitochondria today, and we analyze those two membranes, and we look at the fatty acids that make up the phospholipids in those membranes, what we notice is the inner membrane of the mitochondria has different fatty acids than the outer membrane. They're both membranes, they're both phospholipid bilayers, but they're made up of different phospholipids. The different phospholipids have different fatty acids. So if we analyze the inner membrane of a mitochondrial organelle, the inner membrane has the exact same phospholipids that bacterial cells have in their membrane. So basically, the inner membrane of a mitochondrion is made up of prokaryotic cell phospholipids. Right? Where did they come from? This is a eukaryotic cell. That's one of the evidences for the endosymbiotic theory. How did the inner membrane of a mitochondrion get these phospholipids? Because at one time they were a free-living bacterial cell that got engulfed. Right? That's evidence number 1. Evidence number 2. If we look at the outer membrane of the mitochondria, the outer membrane, and we look at the phospholipids of that membrane, what do we find? Those are phospholipids that are characteristic of eukaryotic membranes. They've got different fatty acids. So in a mitochondrion that we see today, the inner membrane is prokaryotic-like, the outer membrane is eukaryotic-like. Right? Kind of strange for a eukaryotic organelle to have that kind of configuration, but the endosymbiotic theory explains it. Yes? What's that?
What does ATP stand for?
Okay, what's the ATP stand for? Get that? That'd be the hydrogen moment. All right, adenosine triphosphate. Okay, we got numbers and stuff. I guess. Yes.
Can you repeat what you said about the evidence?
About what?
What you just said earlier. Just a minute.
It's a lot of things.
Okay.
Be more specific. I'm not reading that shit.
All right. So we're talking about the.
Of the inner membranes?
Yeah.
All right. So the inner membrane has the phospholipids that are prokaryotic-like, right? So we find different phospholipids in prokaryotic membranes than we find in eukaryotic membranes. So the inner membrane of a mitochondrion has The outer membrane has eukaryotic-like fatty acids on its phosphates. Right? So those are two strong pieces of evidence. Let's go one step further. Another piece of evidence that supports the endosymbiotic theory. Let's go back to this bacterial cell. Does that bacterial cell have any DNA?
Yes.
What does it look like? What? It's a single chromosome that is circular. Do we find circular chromosomes in eukaryotic cells? No, right? If we go into a mitochondrion today, we go into one of your cells, and we grab a mitochondrion and we pull it out, we open it up and we look inside, will we find DNA in a mitochondrion? And the answer is yes, there's DNA in mitochondria. Mitochondrial DNA. Every one of your mitochondria has DNA. What does it look like? It is a single piece of DNA and it is circular. Do eukaryotic cells have circular DNA? Yeah. The mitochondria do. Where'd it come from? Came from, evolutionarily, from that bacterial cell that was originally engulfed. That DNA is still there in the mitochondria. So mitochondria have their own DNA. So, if that evidence is there, all mitochondria show that, right? All mitochondria have prokaryotic-like inner membrane and eukaryotic-like outer membrane. They all have a circular piece of DNA. So, if we reached into one of your cells and we grabbed a mitochondrion, we took it out, we got rid of the outer membrane and set it free, would it live? Could we reverse this, in other words? Would it live? It's got DNA, it's got basically a bacterial cell now. Would it live? Just say this for a quiz question. This is a great quiz question. At least from my standpoint, I didn't know the answer. What do you think?
Technically, wouldn't it, because it has its like own prokaryotic properties, but it's been in that host for so long, wouldn't it struggle?
Why would it struggle?
Why would it struggle? It's not getting the carbon compounds from.
Okay, let's say we set it free and we put it into a sugar solution.
It would still be getting from that then. Okay.
So will it live?
Sure. I don't know. It's kind of a trick question.
It's funny. When I ask that question, someone says sure, and I just stare at them for a moment, and they go, I think. I stare at them a little bit longer, and they'll say, I don't know.
I don't know. I really don't.
Things go from yes to maybe to I don't know to please give me the answer because I don't want to do this anymore. All right. So the answer is no. Actually, you're right. They would not survive, and here's the reason why: is even though the mitochondria of today's cell have a circular chromosome, they're missing a few of the genes they would need to survive as an organism. They're missing a couple of, maybe more a couple, probably about 3, 4, 5 genes that they would need. So what happened to those genes over time? They didn't just go away. Those genes that used to be here in the mitochondria, you know where they are now? They're in the nucleus. They're in the chromosomes of the nucleus. So the nucleus has kind of absconded with a few of the genes. It's kind of like, okay, you are now my right? I'm taking a couple of your genes. So if we set that mitochondrion outside, it would not survive because it's missing a few of the genes. Some of those genes have migrated to the nucleus. So it truly is now an organelle. It's not an endosymbiont. It is truly an organelle because now it's dependent upon the host cell. Yes?
The mitochondria is missing genes.
Say it again?
What are you saying that the mitochondria is missing genes?
It's missing some of the genes it would need to survive as an organism. Okay. All right, so if this is the mitochondrion, the other double membrane organelle we talked about was what? The chloroplast, right? The chloroplast. So what's different with the chloroplast than the mitochondria? It still has 2 membranes, but what else is different about it? It has chlorophyll, right? It's green. The chlorophyll photosynthesizes, right? So where did it come from? Well, the same theory explains the origin of chloroplast. The only difference is the origin of the mitochondrion is because of a bacterial cell is engulfed. The origin of the chloroplast is a photosynthetic bacterium that can undergo photosynthesis. And there are a lot of green bacteria that undergo photosynthesis. So it's the same thing. Look at the evidences. There are two membranes. Inner membrane of the chloroplast, prokaryotic life. The outer membrane, eukaryotic life. Do chloroplasts have DNA? These questions, 50-50 chance. Yes, they do have DNA. What's it look like? Take a shot. It's a circular piece of DNA. It's a bacterial chromosome. But again, the chloroplast is missing a few genes. It's missing a few genes that have migrated over to the nucleus. So these are genes that the chloroplast doesn't need to function as long as it's an organelle. But if you try to set it free, it wouldn't have it all. It would still need those genes. Yes?
So, it becomes an organelle when they depend on each other, but if it's just like a
Well, it's an organelle now because it was originally engulfed as a free-living bacterial cell. It got engulfed, and the nucleus kind of, quote, stole some genes out of it, so now it's captive. And now we refer to it as it's actually an organelle because it could not function as an organism anymore. But it's got everything it needs to function as an organelle. But it can't function as an organism. Yeah.
So before this happened, was it possible for like cells creating enough energy to actually make a large organism?
Say that again, man. I'm not sure what it is.
Before this happened, would it be possible for cells to actually like have create enough energy to like hold a large organism?
So like a multicellular organism? This probably occurred evolutionarily before multicellularity. Or if we did have multicellular organisms, they were very, very small. Maybe a dozen cells. Not the organisms we see today. So this, in most cases, this is probably a unicellular organism. So this is a single cell eukaryote that engulfed that. Still was maintaining, and then multicellularity evolved many years later. Yes?
What did you say the difference between the bacteria was for chloroplast?
In fact, this is a bacteria that's not photosynthetic. And so what gave rise to the chloroplast would be a photosynthetic bacteria, one that could undergo photosynthesis. And those bacteria exist today. There are green bacteria that photosynthesize, that are prokaryotic. They're still in existence today. All right, so that's the endosymbiotic theory of organelle origin. All right, so in the, what we got, 2 minutes left. In 2 minutes left, we're talking about, introduce the cytoskeleton. We'll finish this up on 5. So the other thing we're going to talk about is what's called the cytoskeleton. Okay, so the term, the prefix cyto means what? Cell, right? We're talking about cytoplasm, meaning cell body, right? So cyto means cell. So what is skeleton? What does that refer to? Structure and support, right? Okay, so this table 7.2. Do not memorize this whole table. If you want to, you can. All right, I'm not going to say you don't have to. What I want you to know is, this. I want you to know what are the components of the cytoskeleton. There are 3, right? There are actin filaments, intermediate filaments, and microtubules. And that's in increasing size. Okay, so microfilaments or actin filaments are the smallest. Intermediate filaments, hence the name, they're the intermediate size. Microtubules are the largest. You can see about dimensions here. This is 25 nanometers, and that's 7. So there's one small. I don't want you to worry about this part. I want you to know what are the components of the cytoskeleton, those 3 components, relative size, and then what do they do? Now you look at this and you say, man, that's a lot of stuff to memorize. But I'll make it really easy for you to understand this. Think of your skeleton. What does your skeleton do for you? Support is one, and what else? What? And movement, right? I'm able to walk back and forth. You walk into the room because of your skeleton. Right? So support and movement. Now, if you go down this list, you can take everything that's on this list, and you can pretty well categorize it as support or movement. Right? For example, maintain cell shape. What's that? That's support, right? Maintain cell shape. Move cells via move. Well, that must be movement, right? Divide animal cells into 2. That's movement. That's moving the cytoplasm, right? Separating things. Move organelles. Well, there's that move word again. Movement. Get down here. Maintain cell shape. That's support. Anchor the nucleus. What does that mean? Support. Holding the nucleus in a particular position, right? So as you go down here, all these functions do what? movement or support. Now, one qualifier on here, actin filaments and microtubules do both, movement and support. Intermediate filaments are not involved in movement. Notice those two things here, they're maintaining cell shape and they're anchoring the organelles in movement. That's all support. Intermediate filaments are not involved in movement. On Friday, we'll talk about how these are involved in movement and where they're going. Alright, every week, we'll see you on Friday. Yes, sir.